special guest key speaker, Professor Dr. Deepak Kumar, Professor sir. It is now time to honor our esteemed guest of honor, Professor Dr. H.S. Hum, sir, Vice Chancellor, Surajmal University. We are deeply grateful for your presence and invaluable contributions. I now request Pension and Executive Director, Sri R.J. Chaudhary, sir, to present token of gratitude to our esteemed guest of honor.
सेंटर कर लेगा ना।
अरे हाँ कल आओ तो
पोस्ट कंप्लीट किया है अभी इसलिए यस यस हाँ जी
वैसे जब पढ़े ना बंद
Thank you so much, sir. That was a significant part of our conference. Please join me in welcoming the convener of our conference, Dr. Manoj Visc, sir, Principal, Devastini Vidyapeeth College of Pharmacy, to the stage to deliver the welcome address.
A warm and pleasant good morning to all present here. On behalf of Devastini Vidyapeeth College of Pharmacy, I take the honor to welcome our esteemed chief guest, Professor Dr. D.S. Rawat sir, the Honorable Vice Chancellor of Puma University. Our special guest, Professor H.S. Dhami sir, the Vice Chancellor of Surajman University, and our key speakers, Professor Deepak Kumar and Professor Gupta sir. One more speaker is with me online, and I just announce the name. Professor Rajiv Dahiya, he will join us online from West Indies later on. And today we all are here together to witness the part of the conference, one day Indo-Caribean International Conference on Global Aspects of Drug Design, Pharmacology and Pharmaceutical Sciences. My heartfelt gratitude to our Honorable MD, Sri Manish Singh Kosiari sir, and our Chief Advisor, Professor N.S. Bhandari sir, our Executive Director sir, Sri Ramesh Chaudhary sir, and Camper Director, Sri C.S. Mehta sir, for giving us all the necessary support for organizing the event. Along with our collaborators, APP West Indies branch, APP Uttarakhand and Solan branch, and I would like to extend my deepest gratitude to UCOST and Pitbull for supporting our conference. The objective of the conference is to provide opportunity to share the knowledge, foster collaboration and inspiring new ideas that will save the future of pharmacy world. Also, it will highlight the recent updates in the field of new drug discoveries and the role of drug designing, and how drug design may provide the tailor-made concept of drug to each individual in the coming future. I am sure this conference will bring all brilliant minds to a common platform, share their expertise area of work for the new growth and upliftment of pharmacy fraternity. This conference will make its foray into new fields of drug design, pharmacology and pharmaceutical sciences. Once again, I welcome all the participants and truly say, without the participation of youth, this would not be possible. Your presence here signifies a commitment to pursuing the boundaries of science and making a lasting impact on our body. Thank you very much.
Thank you so much, sir, for your inspiring words. To share a few words of wisdom and to bless us with our inspiring thoughts, I would now like to invite our esteemed chief guest, Honorable Vice Chancellor of our University, Professor Dr. Divan Singh Rawat, to the stage. Please join me in welcoming with a warm round of applause.
Thank you. Sri M.S. Koshari, Professor N.S. Nandar, Professor Dhami, Chaudhary Sir, other distinguished guests, and dear students. Let me thank Professor Bhandari first because he forced me to come here this morning. As I had so many meetings lined up, but I thought let's visit the campus and see what kind of campus it is. I'm very, very happy to be here and thank you for your kind invitation. ये जो कॉन्फ्रेंस का जो थीम है, आफ्टर बिकमिंग वाइस चांसलर एक चीज बड़ी कॉमन हो जाती है, आप किसी सब्जेक्ट के एक्सपर्ट हो न हो, आपको हर फोरम में बोलना होता है, एंड दैट मेक्स लाइफ वेरी डिफिकल्ट एज ए साइंस स्टूडेंट। इससे पहले जहाँ भी जाते हैं अपनी साइंस जो है उसको बोलते हैं, उसके तैयारी की जरूरत नहीं पड़ती है। बट दिस कॉन्फ्रेंस इज एज सच दैट ये मेरा अपना डोमेन एरिया है, सो मुझे यहाँ पर आकर के ज़्यादा अच्छा है। When we talk about drug discovery, and chemistry एक ऐसा साइंस है जिसके बगैर कुछ भी संभव नहीं। I may be pardoned by Professor Dhami because he is mathematician. So chemistry के बगैर मुझे नहीं लगता है कि कोई चीज़ possible है। Talking to you, you are receiving signals, जो भी चारों तरफ आप देखते हैं chemistry. Let me tell you just historical fact how this branch has been. अगर हम आप पुराणों में जाएं तो उसमें ये बोला जाता था कि हमारे जो ऋषि मुनि थे उनकी एज 1000 साल है। बट ऑर्गेनिक केमिस्ट्री जो ड्रग डेवलपमेंट एरिया जहाँ से शुरू होता है उसकी जो स्टार्ट हुआ 1828 से हुआ, जब फर्स्ट ऑर्गेनिक मॉलिक्यूल was discovered by Ohl in 1828. And if you read the literature, Ohl was not a chemist, he was a medical doctor. So जो medical student होते हैं, जो engineering students होते हैं, उनको subject जैसे chemistry पढ़ना होता है. And at that time, Professor Gremlin was one of the professors with whom he started his classes. As a teacher, हमारा काम ये होता है कि कौन बच्चा कितना अच्छा उसके हिसाब से आप उसको गाइड करते हैं। And Gremlin motivated him to join the classes of J.J. Purchase, one of the famous chemists of that time. And Purchase की जो hypothesis थी वो ये थी कि organic compounds को आप lab में नहीं बना सकते, ये living organism बना सकता है। And when Wohler started working with him at the age of 28, he discovered the first organic molecule and proved that organic molecule can be synthesized at level. So जो उनके famous boss थे उनकी जो hypothesis थी उसको discredited किया। But if you look at the history after that, 1828 में ये हुआ, उसके बाद 1874 दो मेजर डिस्कवरी केमिस्ट्री, एक बट दो लोगों की थी जिसको हम स्टीरियो केमिस्ट्री कहते हैं, 3-dimensional structure of the compound. So these young men, one was French, another was German, said कि कार्बन टेट्राहेड्रल, उस समय के जो फेमस केमिस्ट थे कोलबे, हरमन कोलबे, he commented that this is a childish fantasy. 1874, and nobody paid attention कि उस टेट्राहेड्रल कार्बन का हमारी लाइफ में क्या इफेक्ट हो सकता है, until 1957 to 64 जब एक ड्रग मॉलिक्यूल को डिस्कवर किया गया जिसका नाम थैलेडोमाइड। Thalidomide was licensed to 30 countries across the globe. And उसका काम था थैलेडोमाइड, उसको वंडर ड्रग का नाम दिया गया. And this was very effective for the treatment of morning sickness of pregnant women. But after 7-8 years, when data were collected, it has a cost of recurrence. In a sense, कोई भी pregnant woman उस समय जो जिसने इस ड्रग को दिया, more than 10,000 babies died. And those who survived had permanent disability. So when the structure of thalidomide was looked at, ये पाया गया उसमें एक कायरल सेंटर है. कायरल सेंटर का मतलब है कार्बन की चारों वैलेंसीज अलग अलग रूप से जुड़ी हुई. जो 1874 में डिस्कवरी थी टेट्राहेड्रल कार्बन वो 1964 के आसपास हुआ. Then the US FDA came in picture. रेगुलेशन बने कि आप किसी भी ड्रग को इस तरीके से सेल आउट नहीं कर सकते until unless you prove the target enzyme. Enzyme क्या होता है? आपका हैंड ग्लव्स जैसा, राइट? So you have a right hand and right hand glove, put it to the counter. But अगर right hand gloves में left hand को आप डालेंगे वो discomfort होगा. And this is what drug enzyme interaction is. But if you go back little bit back than this, जो पहला मॉलिक्यूल था जिसको आप एज अ ड्रग बोलते हैं, जिसने पूरी मेडिसिनल केमिस्ट्री को रिवोल्यूशनाइज किया, वो पेनिसिलिन 1940 के आसपास. एंड इफ यू आस्क अ जनरल क्वेश्चन टू एनी ऑफ द स्टूडेंट्स कि पेनिसिलिन की डिस्कवरी किसने की, एवरीबॉडी विल से. अब जैसे हम लोग अगर एक जनरल क्वेश्चन जीके में डालें कि सन टेम्पल कहाँ है, एवरीबॉडी विल टेक पूरा. हमारे पास भी अपना एक जो 900 साल पुराना, क्योंकि हम उसके बारे में नहीं जानते हैं, उसके बारे में हमने पढ़ाया नहीं, किसी को दिखाया नहीं, इग्नोरेंट. मुझे अभी 7-8 साल पहले पता यहाँ पर भी सही. So if you read the story of penicillin discovery, 1942 या 41 में वो लाइसेंस हुआ मार्केट. उसका रेसिस्टेंस 2 साल पहले रिपोर्ट हुआ. डिस्कवरी से पहले कि ये ड्रग रेसिस्टेंट इस बैक्टीरियल स्ट्रेन के लिए रहेगी, तो पहले मार्केट में. But penicillin discovery, as we all know, was an accidental discovery. But उसके ज़्यादा ना हम बच्चों को पढ़ाते हैं, ना हम लिटरेचर को पढ़ते हैं, क्योंकि किताबें हम को लिखनी है. तो जो शॉर्टकट होता है वो मेथड हम अपनाते हैं. हमने लिखा कि पेनिसिलिन की डिस्कवरी ऐसे. But if you look at the real history of penicillin discovery, he published the first paper Fleming in 1929 in Journal of Pharmacology. After that, he did not publish a single research paper on penicillin. In 1936, Edward Florey and Kim came together and they thought कि इसका इतना बढ़िया पोटेंशियल है, ट्राई करते हैं कि इसको आइसोलेट कैसे करें पेनिसिलिन. But penicillin production was very difficult. The fourth person came in picture. उसने उसको जेनेटिक इंजीनियरिंग के थ्रू उसकी यील्ड बढ़ाई. But जब नोबेल प्राइज मिला, 3 people got the Nobel Prize. Fleming, Florey and Edward Fleming. The fourth person जिसकी वजह से इतना ज़्यादा प्रोडक्शन हुआ था, that person remained unnoticed in the literature. Fleming got 128 awards and recognition for penicillin discovery. The fourth person got only 2. And Harvard, Cambridge University में non-medical person को पहली जो TSC मिली थी 800 साल के इतिहास में, that was that person. And the story says अगर वो fourth person अगर नहीं होती तो पेनिसिलिन उस स्टेज पर नहीं होती जहाँ. जब ये नहीं था तब लाइफ कैसी थी, जब हमारे पास ड्रग्स नहीं थे. 1828 1900 average age of human on earth was 23.5 years in India, जो आज के दिन 66 से ऊपर. How that possible? This was made possible because of the branch of science what we call chemistry. Because we are the people who can make the carbon, like architect building खड़ा करता है. So we have that, no? We can break a carbon bond and we can make a carbon. So making and breaking this carbon bond is crucial for this branch of science. Nature जो का जैसे आप plants को देखेंगे, most of the compounds plants बनाते हैं. हम कहते हैं हमारे उसमें कोलेस्ट्रॉल है, बैड कोलेस्ट्रॉल. What is cholesterol? Cholesterol is a organic compound. But अगर इसी कोलेस्ट्रॉल को आप लैब में बनाना चाहते हैं, तो you have to heat it up, maybe 100 degree centigrade for some reactions and some reaction at minus 80 degree centigrade. But हमारी बॉडी में उसकी जरूरत ही नहीं. Ambient temperature जो आपका एटमॉस्फेरिक टेम्परेचर उसको बनता है. हमारे पास जो सॉल्वेंट है वाटर, and most of the organic reactions cannot be performed in water. So this is the beauty of nature versus what we do. But अगर हम ये कहें कि old practice की बहुत अच्छा है, that may not be that correct, because at the end of the day you need something cure. हम इस चीज़ को अप्रिशिएट इसलिए नहीं कर सकते क्योंकि हमने वो दिन नहीं जब पेनिसिलिन नहीं था, कोई भी बैक्टीरियल इन्फेक्शन होता था, आदमी की डेथ सर्टेन हो. बट वी स्टार्टेड अप्रिशिएटिंग वैल्यू ऑफ दिस ब्रांच ओनली आफ्टर द कोविड. इफ यू रिकॉल जब कोविड हुआ कि कोई भी ड्रग या वैक्सीन नहीं थी, तो पूरे जो सिटीज लॉकडाउन हुआ. व्हाई? बिकॉज देयर वाज नथिंग टू क्योर that kind of virus. So today this branch has progressed really well. And बच्चों को ये बोलना चाहूँगा, आप जब आपने ये सब्जेक्ट को चूज किया है फार्मेसी, so you must have some idea why you have chosen. Number two, there is no dearth of jobs in the country. Let me make this straight. Because we go in so many committees. The problem is the quality of the individual. We have countless number of graduate, postgraduate, doctorates. But when it comes to the quality is not. So we need to focus on quality education. We need to be very careful what we are aiming for. कि आपको कल को क्या बनना है. एंड आपको स्टडी करनी है, होलिस्टिक स्टडी करनी चाहिए. ये नहीं कि आप एक सुपरफिशियली चीज़ों को करें. हमारे अपने एजुकेशन पॉलिसीज़ की कुछ खामियां रही जिसकी वजह से हमको शायद वो तरक्की नहीं मिली जो मिलनी चाहिए. Now today there are discussion in the parliament only for few things which I feel is not going to help any. If we keep on talking on this issue, apart from the quality, we never talk about the quality, we talk about other issues. But if you go back, when we talk about the caste, religion, all that, just read the classical textbooks. हिंदुस्तान का अगर हिस्ट्री को पढ़ना है, उसको समझना है, तो क्लासिकल बुक है वो इंडिका करके, जिसको लगभग 2300 ईयर पहले जब चंद्रगुप्त मौर्य का शासन था इस कंट्री, उसमें एक फिलोसोफर थे मैग्नेटस. ही विजिटेड इंडिया एंड ही स्टेड हियर फॉर 7 years and he wrote a book called Indica. Unfortunately the book was lost now, but its abstracts are available. Based on these abstracts, the foreign authors and Indian historian has made their narrative. उस बुक में हिंदुस्तान जातियों का वर्णन 7. These 7 castes are not those which we know today. वो नहीं, उसमें shepherd, उसमें contractor, उसमें lawyer. So he said this is the society which is completely different. But the caste, हमारे अपने धर्मों तो वर्ण की बात की, हमने 4 वर्ण की बात की, हमने कास्ट की बात की. Today we are so choked out कि इसके अलावा शायद दूसरी चीज कोई है ही. The caste word is not even the English word. It's a Portugali word. The first time in India that was used in 1931 when the census was done. And at that time, the Britishers mentioned 4114 castes, which is now more than 6,000 or something like that. कहने का मतलब ये है, आप जिस भी सब्जेक्ट को पढ़ते हैं, स्टैंडर्ड टेक्स्ट को पढ़ने की बहुत जरूरत है। नंबर बी एनालिटिकल एज अ साइंस स्टूडेंट। साइंस की डिस्कवरी कैसे होती है? साइंस की डिस्कवरी तब होती है जब आप क्वेश्चन रेज करते। इफ समथिंग सेज दिस हैपेंस लाइक दिस, यू मस्ट हैव द कैपेबिलिटीज। व्हाई? तो क्वेश्चन आस्किंग जो कैपेबिलिटीज होती है, दैट लीड्स टू नोबल डिस्कवरी। सो एक चीज को आपको ये ध्यान रखना। दूसरा, जो हार्ड वर्क है एंड इंटेलिजेंस, इन दोनों का आपस में कोई भी रिलेशन नहीं। द इंटेलिजेंट पर्सन may not be successful in life. Let me tell you one thing. The person who is hard working will always be successful. With last one quote, I will just stop my speech because I have put an online meeting. 1987 में, आप क्रिकेट सभी लोग देखते होंगे, 87 में मैच हुआ था हैदराबाद, इंडिया वर्सेस ऑस्ट्रेलिया। Brad Hawk was a young chap at that time. Bowled out Sachin Tendulkar, who was playing at 50 odd runs or so. तो उसके लिए वो सबसे बड़ी अचीवमेंट थी, because a young person bowled out the famous batsman of the world, Sachin Tendulkar. In the evening, he went to the dressing room of Sachin Tendulkar to photograph. He said कि मेरे जिंदगी का सबसे अच्छा दिन है क्योंकि मैंने आज आपको बोल आउट किया। Can you please give me your autograph? Sachin signed on that photograph. He wrote a statement on the top. He said, "This day will never come in your life again." मैं कहा कि आपके जिंदगी में ये दिन दोबारा नहीं आएगा। फाइन स्टेटमेंट, हम भी बहुत सारी चीजें कहते हैं, लिखते हैं, बोलते हैं, but what happened after that? Both Sachin Tendulkar and Brad Hawk played 17 matches after that in next 7 years. Brad Hawk not bowled. क्या मतलब है इसका? इसका मतलब ये, जो कमिटमेंट सचिन ने अपने लिए किया था कि मैं ये करूंगा, he must have practiced up to that level. He fulfilled his own promises. So make your promises for yourself, not your parents, not your teachers, not your friends. Periodically review it, that what promises you have made, whether you are doing the same kind of efforts to achieve the promises that you have made yourself. If you do that, let me tell you one thing, you will be the most successful person in your life. With this, I thank the organizer for the success of this wonderful program. Thank you very much for having me. Thank you.
Thank you so much, sir. So before you leave the stage, we would like to present you the memento. For that, I would like to invite Shri Manin Koshari sir, Professor N.S. Bhandari sir, Mr. R.C. Chaudhary sir, Professor Dr. C.S. Mehta sir, and convener Professor Dr. Manoj Bis sir. Please come forward and present the
Shiva? Further? ठीक है।
Thank you so much, sir. Sir, chief patrons and patrons, please remain in the stage. Patrons and chief patrons, sir, please remain in the stage. I request our convener. Thank you so much, sir. stage. They will be sharing their gratitude message regarding conference with us today.
Good morning everyone present here. It is an immense pleasure and honor for me to stand before distinguished gathering of eminent personalities, scientists, researchers and professionals around the world on one-day Indo-Caribean International Conference in global aspect of drug design, pharmacology and pharmaceutical science in collaboration with Association of Pharmacy Professionals being organized at Devsili Vidyapith, College of Pharmacy, Lalpur, Uddapur. Conference offers a wonderful platform for interaction and exchange of scientific initiatives and ideas. It also provides great platform to further explore the ever-expanding horizon of academic research. So I wholeheartedly welcome the delegate across the country. It will be great to hear from our keynote speakers in the field of pharmacy. We are also eager to hear from our delegates about their ideas and innovations. I hope the upcoming scientific session will be fruitful to all the members present in the conference and will inspire to all of you. The program includes keynote presentations, lectures, oral and poster presentation will mark and enhance the scientific knowledge toward them. I hope this premises will inspire all of us to sharpen our vision and achieve our individual goals, while further enabling you to discuss critical issues related to your practice development. I, Ramesh Chaudhary, Executive Director, Devsili Vidyapith, extend my sincere thanks and deepest gratitude to our Chief Guest, Professor Diwan Singh Rawatji, Honorable Vice Chancellor, Kumaon University, Nainital, who has accepted our invitation by sparing his valuable time for addressing this conference and giving blessing to our students and participants. I also extend my sincere thanks to our guest of honor, Dr. H.S. Dhamiji, Vice Chancellor, Surajmal University, Kicha, and eminent speaker, Professor Gaurav Gupta, Professor Deepak Kumarji, and Professor Rajiv Dahiyaji, and members of our managing committee, particularly Sri Manji Kushalji, Managing Director, Sri Bhandari our Chief Advisor, Sri C.S. Rawatji, Sri C.S. Dr. C.S. Mehtaji, for making their presence in this conference. Last but not least, I would also like to thank all the scholars who have taken time to prepare submission for this conference. We are honored that you choose this conference as a platform of disseminating your valuable research outcomes. Thank you for your presence and participation. I am grateful to Dr. Manoj Bish, convener of the conference will prove a great milestone in the field of pharma sector. Thank you.
Thank you so much, sir, for your valuable words. We will now take a short break for tea. Please take this opportunity to network, and we will reconvene for our next session at 12 o'clock. Thank you so much. For delegates and distinguished guests, please approach for the ground floor, and for the participants, you have to go for the canteen. So here we come, the start of our scientific session. To continue the conference, we have the privilege of hearing from Professor Dr. Gaurav Gupta. Dr. Gaurav Gupta is an esteemed adjunct research professor at the Center of Medical and Biohealth Sciences Research, CMBHSR, at Ajman University, UAE. He has a prolific academic background, having served as a professor of pharmacology at Suresh Gyan Bihar University, Jaipur, and holding adjunct professorships at Graphic Era University, Dehradun. Dr. Gupta has earned his doctorate from Pacific University, Udaipur. With over 13 years of teaching and research experience, he has gained extensive international research exposure in laboratories across Malaysia and Australia. He is recognized among the world's top 2% of scientists by Stanford University for the past 4 years. He is a prominent figure in the academic community with numerous peer-reviewed publications, book chapters, and presentations at national and international conferences. Dr. Gupta has made remarkable contributions to the field of pharmacy with over 600 research papers and reviews published in SCI Index journals. His research primarily focuses on molecular and biochemical pharmacology, including phytochemistry, respiratory diseases, cytopharmacology, and cancer biology. Now, I would like to call upon on stage Professor Dr. Gaurav Gupta sir for his presentation. Please welcome him with a huge round of applause.
Hello everyone. A beautiful, pleasant good afternoon to all of you. First of all, I would like to congratulate the Devasli Vidyapith management for organizing a one-day of global international conference involving the drug designing, pharmaceutical sciences and biological molecules. So at today's conference, I am Dr. Gaurav Gupta going to discuss about the role of artificial intelligences in the pharmaceutical sciences. This artificial intelligences, this is a very common term. Nowadays, if we are going to the social media, if we are going to the Facebook, if you are going to the newspaper, everywhere you are hearing this term artificial intelligence. So, as a student, in future, in coming era, you have to be compete with the artificial intelligences in your research, in your job sectors. So, you should be familiarized with the artificial intelligences. You should learn about the technologies where this artificial intelligence is involving in the pharmaceutical sectors, how they are playing significant roles in the pharmaceutical sectors. You should know about all these things. You can see your future scope in artificial intelligences. So we'll see what is this artificial intelligences, what are their significant roles in our regular life, what are their roles in the pharmaceutical sectors, what are their challenges, and how can we make our career in the artificial intelligences in the pharmaceutical sectors. All these things we will discuss in our this talk. So in this talk, we are going to discuss about the artificial intelligences. What is artificial intelligences? What is application in our regular life? What are the applications in pharmaceutical challenges, pharmaceutical sectors, challenges in the pharmaceutical sectors about the artificial intelligences, and how can we make our scope of the artificial intelligence in the pharmaceutical sectors. All these things we will discuss in our. So first of all, what is artificial intelligence? artificial intelligence, it is a computerized operated machine which is working same like human being. So in a layman language, I can say as a human being how our brain is working, same like that a machine is also working. That is called the artificial intelligence. It's very simple term is there. Machinery, a computer operated or the software operated machineries which are working same like human brain, that is the artificial intelligence. So how they are working? Same like a human brain. For that they require the data, a number of data. That data is calling a algorithms. Algorithms, a number of data in a particular manner they are inserting into the computers, into the software, and then they are learning and they are working same like a human being. That is a artificial intelligence. So what is artificial intelligence? It is working same like a human being. How our brain is thinking in a 360-degree of area in all the prospects, if we are thinking about any one point. So how our brain is responding on any one topic. Same like that, artificial intelligence is a computer are also responding same like our human beings. They having a algorithmic data. On the basis of data, they are learning and they are responding, they are commanding on the particular topic of the information, that is a artificial intelligence. So we'll discuss about some examples where we can see the role of this artificial intelligence in our daily life. So first is the virtual assistants. In our mobiles, either we are using the Siri, either we are using the Alexa or any type of the box system. What these both systems are doing? They are taking the commands from the person and they are responding to those commands. Either you are asking about the new songs, you are asking about any type of movies, you are asking about any type of the news updates, anything you are asking. So they are responding on basis of your command. They are taking the command and they are responding. That is the best example of this artificial intelligence where they are playing a role in our regular life. So the first example is the virtual assistants where we are using the Siri, we are using the Alexa, we are using the Google or many other type of the dot systems are there which are taking the command from us and they are responding. That's the best example. Second example is the navigation app. While driving, we all are using nowadays the navigation app, whether it's a Google Drive or this one, Google Map or the Waze. So what they are doing? They are helping us. How they are helping us to avoid to drive into the traffic areas. Or they are giving us the exact correct way of any particular destinations. So you can imagine how this artificial intelligence is, they are making our life easy. So we can say what is the basic role of artificial intelligence in our life? To make our life in easy way to different times. So all these things we will discuss in the next slides. Next example is the social media. Nowadays we all are using the social media, whether it is a Facebook, Instagram, Twitter, whatever these things are there. So what you guys are doing? You are just adding your name or your institution name or your company's name and you are getting a list of your friends. So now it's become very easy to find out and to be in contact with your friends. So it's making your life easy. Online shoppings. We all are doing the online shoppings. Amazon, Flipkart, everywhere we are doing the shopping. So what this artificial intelligence is doing there? They are giving us the alternative option of the same product in a different brands. So they are helping us to find out our product in a less period of time and the better quality of product they are helping us. So again they are making our life very easy. In a healthcare sector. So the most important sector, that is the healthcare sector, the best example is our digital fit watch. We all are using the digital fit watch. So how they are helping us to easily measure our daily routines, how much calories we are burning, what are our daily routines are there. We can easily measure our pulse rate, we can easily measure our oxygen rate, and other things we can easily measure with the help of this fit watch. So other examples are also there that we will discuss later. Next significant role of this artificial intelligence is in the banking. So nowadays the banking sector is also using the artificial intelligence. They are trying to help us, whatever the amount we are putting in our saving accounts in the bank, they are observing those amount. On the basis of amount, they are always suggesting us to invest those amount in a different type of scheme so that you can utilize your amount whatever is there in your saving account. So that is the aim, making your life easy with the help of artificial intelligence. Next example of this artificial intelligence is the smart home devices. Nowadays in our home we are using the remote control operated most of the devices, or the Alexa operated devices also nowadays we are using in our modern home. With the help of that you can easily maintain your security system in your house, you can easily maintain your light system, you can easily maintain your temperature, can control the temperature of your house. So that is called the smart home devices. Again, that is playing a role by the artificial intelligence, which is making your life easy. Next example is the entertainment. In the entertainment, nowadays we all are using the OTT channels, whether it is Netflix or Amazon Prime, whatever things are there, you can easily find out your the type of movies, what type of movies you want to see. You can easily see the list of those type of movies. So these are the few examples of artificial intelligence which are playing the significant role in our regular life. Now you can think and imagine how this artificial intelligence is making your life easy. So now next we will see what is the need for this artificial intelligence, why it is required nowadays. There is a lot of factors are there which is depend on the basis of that the artificial intelligence is required in our daily life. First is the automations. Automations, everything is now converting into the automatic systems. We human beings, we don't want to use our body systems to do the work. We always working like a smart brain. We are just want to use our smart brain system to work any type of the work, whether it is in a physical or in our home or whether it is in our job sector. So the artificial intelligence is required for in such a way. Data analysis to maintain the data. Either it is in the pharmacy sector, in the healthcare sector or any other sectors. As a human being, earlier we used to do the many type of error in the maintaining the data. So due to that error, we are facing a lot of problems. But nowadays, with the help of artificial intelligence, we can easily maintain any type of data. On the basis of data, we can do the further research. So apart from this one, personalizations, in the personalizations, up to the year 2050, each and every person will get their individual doctor, and that doctor will be calling as a AI doctor, artificial intelligence doctor, who can help you to analyze your gene sequencing, who can scan your gene and they can easily give you the predict on your future causing any type of disease. On the basis of that, they can maintain your lifestyle, they can maintain your life, the diet chart, everything. That is calling the personalization for each and every person having the different type of doctors. So apart from this one, innovation, improved decision making, improved safety, cost reduction, these all are the factors are there for what we require the artificial intelligence in between among us. So how this artificial intelligence is the future of pharma? So this is the future of pharma. As a student, this is the future of yours. If you are become familiar with the artificial intelligence, then only you can survive into the pharma sector market. If you are not learning about artificial intelligence, how this artificial intelligence can be used in your research, then it's very difficult. Because nowadays lot of softwares are there which can easily work and they can replace. So this is a risk or this is a challenge for you. If you are learning with this one artificial intelligence, you can bring this artificial intelligence in your research, that would be good. Otherwise it would be very challenging for you to work into the pharmaceutical markets. So we'll see first of all where these artificial intelligences are playing a role in the pharmaceutical markets. First is the current drug discovery process. Drug discovery. We all know about drug discovery. To find out a new drug molecule. To find out a new drug molecule, bring that drug molecule from the laboratory into the market. That is called the drug discovery. And this time period to bring one drug molecule from the laboratory to the market, it usually taking the 15 years of time. It usually taking the billions and billions dollars of investigations by the companies. So you can think about this one, a huge amount is investing by the pharmaceutical companies. How much amount? A billions and billions of dollars for one drug molecule. And how much period time they require? 50 years of time. And after that also it is not sure that that type of drug will be approved by the FDA. There is a very less chances are there. So if we talk about the probability, out of 100, only 10 molecules are finally approved by the FDA, which are allowing to launch those drug molecules into the market. Remaining 90 molecules are getting failed. So now you can imagine how much amount of money is wasting, how much time period is wasting, how much scientists' time are wasting to bring one molecule into the market, from the laboratory to the market. So for that, nowadays artificial intelligence is operated, lots of softwares are there. So what are those softwares that we will discuss in the later? So that we have already discussed, industry is currently said to spend well over $1 billion for bringing one molecule. And time period they require 15 years of time to bring one molecule to the market. So that is very challenging for the industry to invest huge amount of money, huge amount of time. So that is the first challenge. So now see the applications. So first application in the R&D, in the research and development, where this artificial intelligence is playing a role. First is the drug discovery. In a drug discovery, nowadays lot of molecular docking softwares are available, which are involving in the drug designs, molecule docking, like Schrödinger, Autodose software, discovery software, lot of softwares are there. So how this software are working? In the software, the drug proteins, they can easily analyze by binding the disease genes, particular genes in the of the disease in the body. So on the basis of their binding property, we can easily analyze within a very less time period, like in 2 or 3 days or maximum 5 days, we can easily analyze the therapeutic value of the drug, efficacy of the drug, whether the drug can be used on the particular disease of interest or not. So earlier, whatever the time we were investing, like 10 years or 15 years, now we can easily reduce those times by the help of the different type of software, that is called the molecular docking software, which are helping to analyzing or scrutinizing the molecules for the particular type of the disease. Virtual screening, we have already seen the virtual screening role during the COVID time. In distance, the physicians are sitting on the televisions, and you are taking the consulting with the physicians, and they are giving you the consultation. So this is the virtual screening, predictive analysis, personalized medication, drug repurposing, clinical trial optimizations, drug safety, image analysis. There are the various type of applications are there. In that one, personalized medications that we have already discussed about that one, a gene analyzing technique. So this is a new technique that is called the gene sequencing technique. In the gene sequencing technique, with the help of AI, physicians can analyze your gene sequencing. They can analyze your DNA structure. Each and every person has a different genetic structures. On the basis of genetic structure, they can give you the information, like in future what type of diseases you may get after 10 years or after 15 years. So on the basis of that, they will give you the suggestions also how to improve your health, how to maintain your health to avoid those types of diseases. That is called the personalized medications. Drug repurposing. Nowadays in the drug repurposing, as we discussed, for any new drug molecule to bring from the laboratory to the market, it requires a huge time, 15 years of time, billions and billions of dollars are required for that. So for avoiding those type of issues, nowadays the pharmaceutical sectors, companies, what they are doing, they are using the drug repurposing. Drug repurposing, whatever the existing drugs are available into the market, they are trying to find out the other therapeutic goal of those type of drugs again. So that is called the drug repurposing. And again, for those drug repurposing, they are using the various type of molecular docking software, and that is the AI operated software. So these are the few major pharmaceutical companies which are using this artificial intelligence in their R&D. That is Pfizer, Merck. What they are doing? They are using in the prediction of the drug-protein interaction, identifying the protein drug candidates, and the optimizing drug properties. So for the various applications in their R&D, they are using the artificial intelligences in their research. Other companies are Novartis, Johnson & Johnson, GlaxoSmithKline. These are all the major companies which are using the artificial intelligence in nowadays. So now you can imagine if you are become expert, if you are become familiarized with those type of the artificial intelligences operated software, then you can easily get a job. Otherwise, it's a big challenge for you. So drug development, second application of this artificial intelligence is the drug development. Drug development, we have already discussed for this one, to develop a new drug. So earlier, companies investigate huge amount of time, period, and the money for bringing one drug. But nowadays, a lot of artificial intelligence based softwares are there which are helping us to find out the drugs in a very short period of time. Even during the drug discovery, for the preclinical trials and for the clinical trials also, they have reduced the number of the objectives. With the help of this artificial intelligence, they can easily analyze the major therapeutic safety of the drug with the help of artificial intelligence. Next is the diagnosis. In the diagnosis, earlier, whatever we are using by the X-ray or any other type of the analyzing techniques, those techniques are giving us the superficial information of our body. They are not able to analyze the insider area of the tissues or the deep tissue part of the diseases. But nowadays, with the help of artificial intelligence, they can easily give you the exact information where your tissue has damage, where your bone has damage. They can easily give you the information. So high technologies, AI, machineries are available which can give you the exact information where your tissue, where your bones are damaged. Apart from this one, telemedicine, the example, the telemedicine we have seen during the COVID, the doctors are sitting somewhere, patients are sitting somewhere, and they are consulting. That is the best example. That is personalized medicines. We have discussed about the personalized medicines, the another therapeutic roles of the existing drugs. That is the drug repurposing. Disease prevention. In a disease prevention, majorly the pharmaceutical companies, they are interested to invest their money only in a common type of diseases where the number of patients are more in the global, so that they can easily find out their market. Whatever the rare diseases are there, they are not interested to invest their time and the money in the rare diseases. Why? Because they are not getting the market for those type of rare diseases. But nowadays, with the help of AI technologies, it is very easy for the companies to invest their a less amount of money, a less amount of time in those type of the rare diseases also, like a Alzheimer, like a Parkinson's, and other type of rare diseases. So with the help of this artificial intelligence, they are easily investing their money and time in a very short, less amount. They can try finding out the R&D and the drug developments. Epidemic predictions. During the COVID-19, this is the most common example is the COVID-19. In the COVID-19, it was a very risky, everybody was very like in a very problematic way thinking where the person or the society can become the COVID-19 infections. It is a unpredictable things was there. But nowadays, a lot of softwares are available, a geographical softwares are there. In that software, if you insert the geographical data, if you insert the other type of data, they can give you the information in the last 5 minutes, in the last 5 days, or in the 10 days, which area can become the prone for any type of particular infection. So those type of softwares are also available in the market. The best example of those softwares are ProMap, Blue Dot, Metabiota, HealthMap. These are the software. So in the COVID-19, this Blue Dot software, it played a major significant role in the COVID-19. It helped us to find out the major upcoming prone area for the COVID-19 infection. So now you can see how this artificial intelligence is operated software, how they're making our life easy and safe. Remote monitoring. So in remote monitoring, nowadays there are a lot of instruments, the healthcare instruments are available. By using those type of instruments, there is no need to go to the doctor. Without going to the doctor, you can easily maintain your health data. Otherwise, earlier, every time you used to go to the doctor to find out your BP, to measure your blood sugar level, and other type of the health related data. But nowadays, lot of instruments are there which are with the help of AI, you can easily maintain your health data without consulting of doctors. So this all data, all instruments, we are very familiarized with the BP instruments, we are familiarized with the health fit watches, we all are using this health fit watches. This is a new instrument that is calling the digital pills dispenser. You can see this instrument. This is calling the digital pills dispenser. In case of elderly persons, in case of Alzheimer person, in case of the Parkinson case, the persons who having a very short memory. So those type of persons, they are not able to memorize their dosing time when they have to take the medications, in what amount they have to take the medication. It's very difficult for them to remember. So for those type of patients, this is a new instrument, that is a digital pills dispenser, which is sending the alarm to your mobile that this is the time to take the medication, in which dose and in which amount, so that the elderly persons can easily maintain their dosing times. So like that, there is a different type of the remote monitoring instruments are available in the market. Manufacturing. In the manufacturing also, nowadays, most of the companies, the big pharma companies, they are using the artificial intelligence. Why they are using this artificial intelligence is to reduce the quality, to increase the quality of the drug, to reduce the wastage of the drug, to improve the quality of the drug. So for that one, they are using the artificial intelligence operated machines. In marketing, Sanofi, Pfizer, Novartis, other major companies, they are using the artificial intelligence. How they are using the artificial intelligence in their marketing? We all know about the online pharmacies, 1mg or any other type of pharmacies are there. So what we are doing? We are uploading our prescription on those pharmacies. And these companies, they are scrutinizing those type of prescriptions and they are searching whether these prescriptions having my medications, my produced medications or not. So if we talk about diabetes, so diabetes selling pharmaceutical companies, they are finding out the diabetic patients' prescriptions and directly they are targeting to only particular those type of patients who are taking the diabetic medications. So like that they can easily avoid the other population, other nations. They are directly targeting the particular persons who are having the market for their medications. So like that in the marketing also, most of the major companies they are doing. Drug adherence and doses. Drug adherence and doses we have already discussed in the digital pulse dispenser. Like they are sending the alarm to the elderly persons, alarm to the Alzheimer persons who are having the less memory, and they are sending us the signal to take the particular dose of amount of the drugs in a particular time. So these are the various type of the applications of artificial intelligences in the pharmaceutical sector. So now next, what are the current challenges? Where this AI become a problem in the pharmaceutical sectors? First is the automation. Now we have discussed that everything is getting automated. There is no need of human being. Everything is operating by the software. So this is a big challenge for the upcoming generations. If you are not become familiar with the AI, then you will not get the job into the market. Because AI slowly is replacing all the jobs into the market. So that is a big challenge for you. Responsibility and the accountability. Who will become the responsible if any error comes into any technology? If we talk about the banking sector, sometimes we heard that our data has become hacked by the hackers and they have released our data So by using the AI techniques, no one will take the responsibility, no one will take the accountability. Data protection. So have we discussed data protection? So there is a risk with the data protection. There is no security with the data. A smart persons are using the various type of the software who can breach this AI technology. They can easily leak and insert in your all the banking sectors, can leak your data and use the using for the financial profit. Next is the eradicating the humanities. So all the human beings slowly replacing from the market and overtaken by the this AI technologies. The investment for using this AI technologies in your companies, in your business, it requires a huge amount. So right now, only a big companies, they are using the or investing for the AI technologies in their business. Apart from this one, a small business companies, they are not able to use this this AI technology in their business. Again, there is a risk. All the small companies are overtaken by the big companies. Emotional support. There is no emotional support when we are using the AI technology. Future aspects. What are the future aspects where we can see the artificial intelligences in next upcoming 5 to 10 years? First is the human will not be allowed to drive. All the car will become a driverless. So that is the first things where the things or the car driving or the taxi driver, then everybody will be replaced. You will have an AI doctor. Up to 2050, each and every person will get the personalized AI doctor who can scan your gene. They can easily analyze and give you the information about your diseases, about your future causing diseases. They can easily give you the medication. Your boss will be a bot. In future, you will not see any human resource department anywhere, either it's a university, either in a corporate sector. Everything will be replaced by the dog or the bot systems. What they will do, they will take your interview. On the basis of your performance, they will scan your performance, they will scan your complete information. On the basis of that one, they will hire you for the job. Even if you are not performing, they can fire you also from the job. So everything, hiring and firing, all the jobs, all the replacing by the AI technology. Smartphone will be the top. Up to 2050, each and every home will become a smartphone. Everything will be replaced by the computer or the machinery operator. Nowadays also you can see most of the builders, they are using the Alexa in the making the home. So what this Alexa does, Alexa is maintain the security part of your home. Alexa is controlling the electricity or whatever the electric instruments are there in your home. Everything is regulating or the controlling by the Alexa. So up to 2050, each and every home will be converted into the smart. There will be a robot criminals. So in future, you can see the robotic criminals also. So there is a one best Hollywood movie is there, I Robot. If you have not seen, you can see the I Robot, that is the Will Smith movie is there. There they have shown how the robots can become a criminal and how they will become the harmful for the society. Robot will feed us in the sport. The robots can also participate into the various type of the sports activities, the athletic activities. News will not be written by the human beings. So news, especially now also, if you see most of the content, when you are going to write some thesis or dissertation, we all are using the various type of software which are helping us to improve our English writing skills, which can easily avoid any type of typographical errors or the grammatical errors. So like that, you can imagine like this type of software in future, they can easily replace any type of the content writing. So these are the various type of AI roles. Either it is a positive roles, either it is a challenging roles. So different type of roles are there. So as a student, if you are able to familiarize with the AI, if you are start using the AI applications in your studies, and if you are become familiarized with the AI, then it is beneficial for you. Lot of online softwares are courses are available, which is available free of cost. You can attend those type of software courses, and you can learn how AI can be helpful for you in your research, in your study. So that you can become familiarized with the AI in your pharmaceutical sector. Otherwise, it becomes a big challenge for you, and it's a big challenge for you to survive into the pharmaceutical market. Thanks very much. So I am just acknowledging my shot to the Devasthini Vidyapith, who is organizing this conference in collaboration with the Association of Pharmacy Professionals, APP Society, and Dr. Manoj Bis, who has organized a wonderful conference. These are my research collaborators: University of Technology Sydney, Royal College of Surgeons Ireland, International Medical Universities, and the University of West Indies, LBU Chitara. These are the various of my collaborators to whom I am working in the research. Okay, thanks a lot. Thank you. If you have any queries, any questions, you can ask. You are welcome.
Hello. Very good afternoon, sir. Very right and constructive presentations on artificial intelligence. So, in pharmaceutical sectors, it has contributed a lot, and it has been contributing a lot. So, is there any drug recently approved by FDA which are artificial intelligence driven?
With the AI are playing the roles. They are reducing the cost, they are reducing the waste amount, and they are improving the quality by modifying the different type of the places in the technology. There is no any particular drug.
Thank you very much.
Any other please? Thanks, sir. Thank you.
Thank you so much, sir, for your enlightening talk on role of artificial intelligences in pharmaceutical science sector, their positive side and the negative side. Now, we would like to present a memento and a certificate to Dr. Professor Gaurav Gupta as a token of appreciation. May I request our convener, Dr. Professor Manoj Bish, sir, to kindly do the honors. Dr. Kumar is an Innovation Ambassador for the Ministry of Education, Innovation Cell and ICIC, Government of India. His prolific contributions to the field include over 100 research papers in esteemed SCI journals, 40 plus patents with 5 granted and numerous book chapters and edited volumes published by major publishers. He has been honored with multiple awards including the High Impact Factor Research Award, Research Excellence Awards and the Leading Patent Fillers Award. Dr. Kumar is an active member of various professional organizations including the American Chemical Society and the Indian Science Congress Association. His research focuses on medicinal chemistry, drug discovery, bioorganic chemistry,
Thank you for your nice introduction. Respected Managing Director, ICIC sir, and Professor Professor Ravan, Sir Vice Chancellor, Kumar University, respected dignitaries, good afternoon. I would also like to thank, firstly I also like to thank Professor Manoj B.C. sir and Neeraj B.C. sir for inviting me for this conference. My research topic will highlight what the UG is, the basic research and what the research that we are carried in our lab by our PhD students. My research will be focused on the importance of the natural product in the drug discovery. Here we can see that the medicinal plants, which is the Himalayan region, is rich in the various diversified, where there is various plants is present in the Himalayan region, and it can extend from the Kashmir to the northeastern part of India. You can see 8,000 species of medicinal plants are found, and from amongst it, 1748 are the medicinal plants. And this, if you found, because I am working mainly on the cancer drug developments, and I found out that around there were about 100 active medicinal plants were there, and they were very prominent amongst these plants. These are the different plants which has been found, like the common plant which is available in the Himalayan region, like Suessa chiraita, and it is Aconitum heterophyllum, Valerian jatamansi, it is also called as a muskroot. You can say Rebundi chini. It is used in the gastrointestinal disorders. Chiraita has been used for the antipyretic and antidiarrheal properties. Means the Himalayan region is having a lot of diverse plants which can be used for the different biological properties. And it has not been explored yet, and it is a very rich diversification and hence can be explored. For the UG person, if you are doing for the drug discovery process, it is not a very simple, because it takes a long time. If you see the medicinal chemistry, they are working on the large number of the medicinal compounds, and after they are working on the thousand lakh of the medicinal compounds and the heterocyclic compounds, and after from there they get only a single compound as a lead molecules and for the drug discovery. Before, if you start for the drug discovery process, you should know on which disease you want to study and what are the targets. And after that, if you know what is the target on which, because the disease have different target, different mechanism is there. So you have to work on the particular type of the target. For example, if you are working on the cancer, then there is a different mechanism is like for the cancer, like EGFR is there, mTOR tyrosine kinase inhibitor is there. The PK inhibitors are there, then so that you have to find the target, and then you have to identify the bioassay, which assay you need to form. There is in vitro assay is there. After the in vitro assay can be done on the different cell lines can be used, and after this in vitro assay you have to go for the clinical model can be done. Here, this is the history of the drug discovery. It started in the 1990s. And in the 1990s, people were working on the herbal drugs. Because if you wanted to have the lead compound, this lead compound from where it will come? It will come from the natural sources, and it can be descended from the people, from the traditionally people, from our grandparents. They know many things regarding the natural products. From there we can know that this plant is used for the traditional things. Then these things can be applied for the can be applied for the different disease, so that it can be extracted, it can be isolated, and we can prepare the different type of medicines. Here, after that, there is in the 1980s, it comes the commercialization of the drug discovery was there, and combinatorial chemistry is one of the major fields. In this combinatorial chemistry, here we can synthesize large number of compounds by decreasing the times in combinatorial chemistry. It came in the 1980s. Then robotic and automation technique has also been there, which is also helped in the drug discovery process. This has reduced our time. Even the artificial intelligence is also helping us in the drug discovery process. Next. Here, this is the small molecules. These are the different small molecules. This just started in the 1990s, where the first small molecules was the aspirin. And now you can see the discovery of this small molecule has been started from 1990, and it has been still there. If you can see in the 2023, FDA has released around 34 small molecules. Then you can see that the medicinal chemistry is playing an important role in the drug discovery process. Though there is very much improvement in the artificial intelligence, but you cannot deny the work that is done by the medicinal chemist, because finally the chemist has to synthesize the compound, and it has to be tested on the animals. Next. And here you can see for the R&D expenses, the government also need to give the fund for the R&D expenses. You can see the pharmaceutical industries. Most of the multinational pharma industries, they are expanding in the R&D process, but they are also not expanding as much as what they can. They are spending very little amount. For example, if you see Pfizer, it is expending the profit of only 13.8%. It is spending for the R&D process. For the Novartis, it is spending only 9.9% of its profit. Johnson & Johnson, 11.8%. If you see the top pharma companies of India, they are also spending in R&D activities, but they are very little. They also have to come up, and so that they can also expand in the R&D process. Next. Here, not only the pharma industry, but the educational institute, the universities, they are also contributing for the drug discovery process, and their contribution cannot be denied. Even the 40% of all the approvals. This is the report of the 2019. In 2018, 49% of the R&D was done by the small pharma industries and the small institute universities and the research institutes. Next. Here, for the drug discovery process, it takes about 14 to 15 years for the drug discovery process. As Gaurav sir has told, by using the artificial intelligence, we can decrease these durations. For the drug discovery process, there is you can see that this is 3 to 5 years. It's 5,000 to 10,000 compounds we are finding, and after that, it will only take from the 10,000 compounds. You can see only 250 compounds is entering into the preclinical trials. In the preclinical trials, you can see in vitro studies and in vivo toxicity is determined, and the ADMET, pharmacokinetic and pharmacodynamics, and it takes about 1 to 2 years for this process. After that, from the preclinical studies, from the 250 compounds, only 5 compounds is entering into the clinical trials. These clinical trials is taking about the 3 phases, and these 3 phases is taking about 6 to 7 years. And after that, only one new drug is approved. From this new drug approval, it has to be approved by the FDA. Drug approval need to be taken, and this is taking about 1 to 2 years. Market also, there is a post-market surveillance is also is there. If there is any side effect of the drug is reported, then the physician need to be reported to the clinical trials. Next disease, then you have to find out which disease, which therapeutics. Here, these are the target selection. If you see the target selections, most of the drug is acting on the G protein coupled receptors. If you are studying the pharmacology, you can see the G protein coupled receptor. Most of the drug is acting on these receptors. And not only this, some of the drugs are also acting on the enzymes. Hormones and factors is there, ion channels, gateways are there. If you see the local anesthetic, general anesthetic, they are acting on the ion channels mediated. Nuclear receptors is also the next. Then gene, we have to see which gene is responsible for the disease. Then we have to target. When we are designing the drugs, we have to see on which particular genes the drugs will be responsible or active. And not only the genes, but it's the protein, on which protein the drug is acting. We have to see the protein. It is also called as a proteomics. By informatics, then this this is the artificial intelligence. We are using, sir has already explained. Next. This is the different application artificial intelligence. So not only this is the neuroinformatics, if you are studying the activities or if you are studying about the on which the model will be applied. For an anxiety test where we can use the plus maze test is there.
Is responsible. There can also be done by taking the MRI and CT scan of the animals. Ex vivo studies can also be has been replaced because
Interaction is taken. In the phase 3, we are only the number of the patient is increased, and there in the safety also we study the safety, efficacy, and the comparison with the drug interaction is studied. In the phase 4 is called as a post-market surveillance. Post-market surveillance means after the drug has been launched, and if there is any safety or concern is being reported, then it has to be routed through the physicians, and it has to be reported to the person who is doing the clinical trial. Next. Then this is the different pharmacokinetic models is there for the drug designing process that we need to follow whenever we are determining the drug. Then there is the plasma concentration is needed, drug effect is there, how the drug is doing. Then for any drug designing we should follow the Lipinski rule of bond receptor should be less than 5 parts in box, and should be -1 and +5, so that the drug parts in box, so the permeability and the drug viability should be enhanced. Next. These are the different methods by which you can find the lengthening of alkyl chains. Lengthening of alkyl chain is also necessary. Some of the drug is not able to cross the blood-brain barrier if we are designing a compound for the targeted brain drug delivery system. In this case, what we need to do, we can change the different alkyl chain. If the alkyl chains can be, and it need to be optimized so that it can be able to cross the blood-brain barrier and it can target the particular place of the brains. Next. These are by using the drug design approaches. There are different type of drugs which has been developed, like Relenza, Eluverix. It has been used for HIV proteases, neurominase, and this is by drug design approaches has been developed. Next. You can see these are the different. My students are also working on the different phytoconstituent plants also, and they are doing extraction and isolation and fractionation of this compound. Next, these are the different plants on which my students they are working. Next, here you can see these are also the different plants on which we are working, and this only we are also doing the extractions, and then after the isolation we are finding which type of the chemical it is, and we are synthesizing it in the lab also. Next. This how you can do the extraction process for the UGS scholar. What you need to do, you have to collect the plant. The plant selection has to be done by the botanist. It has to be identified by the taxonomist, and the identification need to be both by the macroscopic, microscopic, chromatographic separation need to be done, and after that authentication it has to be extracted using the different type of the solvents. The solvents can be polar, non-polar solvent, and after that the plant extract has been done. The plant extract simply can be used for the biological target, or it can be isolated, and for the isolation purposes you can use the column chromatography technique. You can use if there is a chiral compound, then you can use the chiral HPLC for the separation of this compound, and the separated isolated compound can be used. But we are seeing in many of the institutes what they are only using the extract for the activity. But if they use the isolated compound, then that will be good one. Next. This is how they are the substrate apparatus. How they are doing that. You have to collect the plant, grind it, mechanically grind, powder it, and then substrate apparatus perform the TLC, and after that identification of this compound not only using the mass spectroscopy, but you have to use the NMR, proton NMR, carbon NMR. You can use it, and you have to identify the compound, which compound is there. Next. इसके बाद I think that the the structure which I have drawn in the camera, you can go, it is not visible. We have synthesized in our lab the different type of plus catechin. The total synthesis of catechin has been done in the lab. But it is not visible, therefore it is very difficult. But so before to proceed for the chemical synthesis, we have conducted the literature review and we have found the different type of chemical activities. The literature review we have done. Next. Here also I have drawn the chemical structure using the camera, but it is not visible. The camera, the structures I have drawn. Next color. We have synthesized large of we have done the total synthesis of the catechin. That is also the plus catechin. Before the synthesizing any compound, we have to follow the retrosynthetic analysis we have to do. The retrosynthetic analysis means we know the final compounds and we are finding the target. We are finding out the starting material. After that, we know the starting material. We can use the starting material to synthesize the compound. By using the starting material, we can synthesize this compound. And after that, after synthesizing large number of compounds, I have synthesized around 15 compounds and followed around 15 to 60 steps and got around 80% yield of this compound. And here we have tested against the different type of the cancer cell lines. This cancer is an abnormal growth of the cell, and if it is not controlled, it can lead to the progression and it can lead to the malignant tumor, and it can finally death can occur. Next. How this is the pathophysiology of the cancer cell. It can be invaded to the. Not only it will not affect to a particular body, it can target, but it can affect to all the parts of the body. Next. Here this is the epidemiology, which is around lung cancer around 1.80 million deaths occur, and this is the highest one. Next. We have tested again the A549 and DAFB against the prostate cancer cell line, FG2 cancer cell line, and we have found out that our compound was showing a very good activity against all the tested cell lines. And after that, we have also done the RT-PCR laser test was also done for the activity. Here we have seen that one day was showing a very good activity, that is 4.8 micromolar. The compound was showing the activity against this compound. And next, this is again the breast cancer cell line. Also, our compound was showing a good inhibitory activity. Next. Again, the prostate cancer also, it was showing its weekend highest growth inhibitory concentration of 5.4 micromolar. Next. Against the hepatocellular liver carcinoma cell line, in this also it is 2.5 micromolecules. Means the same compound was showing a good activity against all the cancer cell lines. Then after the synthesis of the compound, we have gone for the RT-PCR and ELISA also we have done, and we found out the compound was showing a very good activity. And after the part we docked this compound against the EGFR, and the interaction was also showing a good result. Here the docking was performed using the Surflex dock program, and this was done by one of my collaborators in the CBRI. Now he performed this docking studies, and this molecular morphing was also done with him. But now in our lab also we have purchased a work station which has been funded by the IIT Roorkee. We have also synthesized dihydropyrimidine compounds, and this compound was showing a very good result, and this was compared with the 5-fluorouracil. This isoprololeucin is one of the anti-cancer drug, and it is in the marketed forms. And here you can see this one. If you see against the cancer cell line, our compound is better as compared to the marketed formulation, and we have find out for the US patent also. And against if you see against the MCF cell line also, it is showing equivalent standard against the breast cancer cell line also. The activity was comparable with this compound. Thank you. Same. And then we have done the in silico studies of this compound, and we found out it was following the Lipinski rule of 5. The molecular weight was less than 500, and it was following all the rules of the drug design. And we have dogged it, and we find out the interaction was very good against the target, which is the NF-κB kappa receptors. Because if you want to go, because we have to prove your, whenever we have to perform the activity, we have to prove also that which molecular level the drug is targeting, then you need to see it. Finally, we can conclude that we have synthesized the total synthesis of minus catechin was synthesized using dihydroxybenzaldehyde, and then phenazole non-compound is also synthesized, and it is showing a very good activity. Bromine dihydropyrimidine was also synthesized and is very, showing a very good activity. And I realized because the chemical here is not working, I don't know that the laptop is not getting the software. Now I just highlight regarding the IHUB Divya which I am looking after the IHUB Divya, and they have funded me for around 2 crore grant they have given me. And this IHUB Divya is located at IHUB Divya, and I am the spoke for it, and I am the coordinator for it. This is they grant the fund. If you have any innovative ideas, and if you can write it directly to IHUB directly, or you can through the spoke center also, you can write it. There will be a call for the proposal, and this call for the proposal is given to the student. For the UG fellowship is there, PG fellowship is there. For them, they will be funding. Now, what is IHUB? That I will go through next. IHUB Divya is a technological innovation hub which has been sanctioned by the government of India. It has been sanctioned around rupees 135 crore has been sanctioned by the government of India. And this IHUB is a Section 8 company. Means they have to give the fund, and they have to generate the funds, and after that they have to run the company. They have as a company. This is run by the NITI Aayog, which is headed by the CEO of the NITI Aayog. DST secretary is also there. The Ministry of Information and Technology, heavy industries secretary is there. Health Research, Central Ministry Department, members of Prime Minister Scientific Advisor is also involved in this innovation hubs, and different IITs is having a translation innovation hub. Next. Now they are supporting for the different things, not only for the school institute, they are also giving for the entrepreneurships, technology developments, and for the training for the skill developments. If you want to train in your institute also, if you want to train, you can apply the funding through the spokes, or you can directly apply to the routine for the skill development. Then they can train, they will give you the fund for the training purposes, and they can even send the academia and the expert and the industry through society, and they can train for the short-term program also. Technology developments will be there. They will connect with the Indian academy and industry also. They will be entrepreneurship will be there. For them also they are supporting. Next, this IHUB Divya recently they have with the chief ministers of Uttarakhand, they have recently organized one meeting with all the police officials of the Ministry of Home, I think, in Uttarakhand region. They have organized some policies for the police. For them also the IHUB Divya has participated. They are the epicenters for the policymakers. They can design what type of policy. They can also suggest the government what type of policy they need to follow. They can support the research and the innovators. They are not only supporting the researchers, they are also supporting the grassroots, the villagers, for the training purposes. They are supporting the institute by providing various fellowships, various schemes is there for the startup also, for the entrepreneurs. They are investing. If you are having any industries and if you are support for them, they are investing also. For the industries can also apply to the IIM. They are also supporting and they are also connecting to the industries also. This is not only benefit to the single person, this is a benefit for everyone. For the student, fellowship is there, opportunities to work with the innovative policy makers. They can get the fellowships, different fellowship is there. For the PG fellowship is there, postdoctorate fellowship is there, UG fellowship is there. For the class 10th and 12th fellowship is even for them also it is there. For the entrepreneurship startup is there, they can apply. For the scheme called Prayas scheme is there. For the incubation support also they can. Seed support is given by them. Accelerator program is there, that is also given. Technology development and entrepreneurship support. Network platforms, they can also provide network platforms. They always organize some network meeting with the government official. For them, they will be able to connect. They are able to connect with the industries. Now this item has been not only in India, they are not focusing only in India, they are also moving into the international collaboration also. They are also collaborating with the international. They have recently opened in the Uganda also, and they are focusing in the South African regions. Next. This is the technology innovation hub. This is also called as a national mission and funding opportunities, and this is also supporting for the product development in the cyber physical system. For the PG fellowship, it is the 31,000 per month is there, and along with the HRA they are providing. For the UG PG fellowship, that is 8,000 per month and 12,000 per month. They also increases depending upon the budgets. Entrepreneurs in residence fellowship is there, that is 30,000 per month is there. Startup funding. This startup funding is around 20,000, and for our university institute they are funded for 2 crores they have given. Next. These are the different fellowship is there. Pump fellowship is there. That is called as the startup funding. Chanakya fellowship for UG and PG. Skill development program for youth in cyber physical systems is there. Innovations. And this is the Surya University, which is located in the Solan, which is means 1 hour from Chandigarh and 1 hour from Simla. It's a beautiful campus, and I also recently got a grant from the DBT pilot, that is 1.77 crore from DBT Center. For if you have any innovative ideas, if you want to make a product in the related to biotechnology, or if you are developing some small molecules, then UVA Center will provide funding. For this, for the PG scholarship, it is 30,000 per month and a contingency of 3 lakh, and for postdoctoral fellowship 50,000 and a contingency of 5 lakh is there. And these are the students who have got this fellowship. These are the 3 students they have got the IHF fellowship, Chanakya fellowship. And this is my research lab, and one of my students is doing a postdoctorate in Taiwan, and one of my students is in Australia, at University for PhD, and recently one emerging center for excellence was inaugurated. That was also Chief Minister of Himachal Pradesh, Bhupinder Singh Sukhu. He has inaugurated that lab, and I also published a paper in the Nature by which I was awarded by the governors of the Himachal Pradesh, and IST also this year is listed in the top 2% scientists of the world. And I'm also looking after the ISTM. Thank you so much. If you have any questions, then right.
Sir, very good afternoon.
Yes, good afternoon.
It's a very tremendous presentation on natural drug formulation and development. My name is Dr. Shishi Nandi. I am also working in this area in drug design and drug development. In India, we have so many eminent labs like CSIR, DBT, DST, and UGC approved universities, different private institutes, eminent laboratories we have. What are the major barriers for drug discovery research, particularly in India?
I think this question will be given by Rahul sir. Will give more. Sir will give more good answer.
Okay. Sir, please. What are the major barriers for drug discovery research in India?
That's very dicey question actually. When we there are two parts of it. One, the market driven. Another is what is our requirement. In India, if you talk in India, we still have a problem with money. Okay? And then tuberculosis. Tuberculosis is a big problem now. Even the other bacterial infections. But if you look at the market, the pharma industry involvement, the business is not too much to that extent. That's why most of the people want to invest their money on cancer, where the market is worldwide market is huge. Okay? So that's the problem, because from Indian academic institutions, we will need to get a single molecule you could get FDA, okay, except ours one in Parkinson's. So these are the problem, because investment is much more required in this case. So we don't, our pharma industry doesn't invest in this. An academic institution cannot afford. So that's
Sir, thank you very much. And sir, I have one more question. By science reported that by 2050 and 2060, almost in every 2 or 3 family, one member will be affected by the cancer around the world. Due to abnormal signaling, abnormal expression, mutations like that. So when a person is suffering from cancer, that person is taking anti-cancer chemotherapy. But is there, mother gives us birth, feed us, protect us from the attack of dreadful diseases. Is there any natural medicines or natural formulations so that if we follow, if we take from the beginning, we can be prevented from the dreadful attack of cancer? I mean to say, you understood my point, sir, what I said is, if a person is taking any natural, plant extract or other natural resources, so that person will never be attacked by the cancer. Is there any research like that is going on, like, family to family, generation to generation, in a particular family or generation, in that family cancer has not been entered? You understood, sir, my point?
Yes,
this question arised in my mind, and you are, sir, very elegant.
Yes, such I don't, I have no idea, but still you can prevent cancer by increasing your immune systems. Okay, therefore by taking the immune, the natural product which increases your immune system, that will help. That will definitely.
So immunomodulator.
That will definitely help you in preventing.
And how say a person is taking immunomodulators through food, through drinks, means natural food, natural drinks like that you are telling?
That can also be possible, but you can medicine, natural medicine, herbal medicine can be taken for cancer.
Okay, sir. Thank you very much. And you have shown that I have.
Yes.
So for young researchers, young faculties who are working like this,
Yes,
not
only I have, if you wanted to apply through EMA center also, that is supported by the bionest. Okay, for them also I can support. From
no, sir, I am telling young researchers, young faculties who are working in private institutions.
Yes, yes.
They are also eligible to apply?
Yes, yes.
For the grant or fellowships?
Yes, then
for
you can
I
apply.
have?
Yes, for the I have as well as for the EMA center. This form will be open throughout India, not only from the particular state, but throughout the India the wall will be open. You can apply, and one model center will be the center, the IM Sampark or IM Sulli or the Yuga center of one that's particular institute. You can choose that, and that your application will come naturally.
Thank you, sir. Thank you very much.
Not only this is, I am only not, the DBT has also nominated one scientist, will be also the overseeing scientist will be there.
Thank you, sir. Very good information. Thank you very much.
Sir, can I ask one question?
Yes, sir.
Hello. Sir, very good afternoon. My name is Samir Goel. I am from Bhimtal. In your lecture, you mentioned platinum-based products, which were actually being used for anti-cancer activities. And of course, your presentation was basically and totally depending on the plants, the herbs which were being used for anti-cancer activities found in Himalayan region. So I have practically two questions to ask. The very first thing is, I'm actually with a background of 35 years in industry. I've been working with oncology. So this was a question which was arising in my mind. We have been working with cisplatin. We have been working with carboplatin. You mentioned these two drugs. Cisplatin has been considered and still being used to test for vomiting sensation for anti-emetic drugs. Because the toxicity is so high that as soon as the patient gets cisplatin, it starts puking. I heard around 10 years back that researchers actually have been working on aqua lip preparation, lyophilized preparation of cisplatin. And after two of these wonderful drugs, cisplatin and carboplatin, no more platinum drugs are coming. So I just wanted to, I was just curious, is there any other drug from platinum group which is in line or which is being researched for? This is the first question. The second question is on plant-based. We received a wonderful drug used to be called as a medicine, miracle bullet from taxanes family. That is paclitaxel and docetaxel. These are being derived from Texas Bacata yew tree. And its plant actually had been cultivated there in Nepal. Originally the drug had been withdrawn from the bark of the tree, and that is not an eco-friendly process, the tree dies. Researchers actually found to find the active ingredient from the needles of the leaves. So that's why they cultivated in Nepal. But we have the same kind of climatic conditions and ecology here in Himalaya. Is there any research actually have been done on cultivating these plants here in India, in Himalayan region? So these are my two questions. The first is platinum based and the second is Taxus Bacata yew.
Yes, and regarding the cisplatin and carboplatin, I want to use as a standard drug. Okay? Because the drug which we have prepared, that was resembling to that carboplatin and cisplatin. And since I know that it was producing the toxicity, therefore after that we are not preparing any platinum containing compounds. Now we are focusing, therefore in the next slide if you have seen, I am using a 5-fluoroethyletoxin as a standard drug, not as a carboplatin as a cisplatin. It has been replaced. And regarding the cultivations, that I have no idea regarding this cultivation.
Because when I found that that was a very interesting.
I am a synthetic chemist, and because in a university, the student they have their own interests. Some student they will come and they will tell me that I wanted to work on a natural product and particles. Therefore, I have to shift my research based on them, not on my idea.
All right.
I have also certain limitations.
No problem. Thank you so much. Thank you.
Thank you, sir, for your enlightening presentation on process of drug discovery from lead identification to preclinical trials and clinical stage. So, now we would like to present a memento and certificate to sir as a token of appreciation. May I request Mr. Manoj Bhargava sir, organizing secretary of this conference, to kindly do the honors. Now I would like to call upon our event coordinator, Mr. Dheeraj Bish sir, to offer the certificate to sir.
सर प्लीज़ कैमरे में देखिए.
Thank you very much, sir. Now I would like to request all the delegates, dignitaries and participants to kindly proceed for the lunch, and we will meet here again at 2:30 p.m. Thank you. Computer-aided drug design, phytochemistry and synthetic chemistry. Dr. Dahiya is also a lifetime member of several prestigious organizations and has an impressive academic record with over 79 publications, 16 book chapters, 3 patents and more than 150 abstracts and 100 posters and many more. May I now request Professor Dr. Rajiv Dahiya to kindly start his presentation.
Hello. अभी आ रही? Okay. I can start the presentation?
Yes, sir.
Okay. My slide is changing. No, my slide is changing. Okay. I am audible? Yes, sir. Am I audible? Yes, please. Transparent. Thank you. So, first of all, good afternoon from Trinidad and Tobago, and I would like to thank the management of the hosting college to give me the opportunity to deliver lecture during this Indo-Caribean International Conference. I would like to apologize. I could not be present face to face due to the emergency conditions. So, by Topical, today discussion is natural cyclopeptide molecules with tremendous biointerest. In this presentation, you will see my 18 to 20 years of experience in the peptide synthesis. My colleagues and students can take the note where they can attend with me for the cyclopeptide related project, and we will educate each other during the continuous session. So this particular conference is being organized in the commemoration of World Lung Cancer Day. So it is annually celebrated on 1st August, and the purpose is aware about the lung cancer and its impact on individuals and communities worldwide. According to World Health Organization, the lung cancer is the leading cause of cancer-related death worldwide in both men and women. Some tips I would like to suggest to help improve lung health. We should avoid smoking, as it is the leading cause of lung cancer. We should limit exposure to outdoor air pollution, industrial chemicals. We should engage in regular physical activity such as walking, jogging, swimming. We should consume a balanced diet rich in fruits, vegetables and whole grains. Some other tips are: we should incorporate deep breathing exercises. We should drink plenty of water. We should ensure up to date with vaccination, and we should have a healthy weight. Before going to the national cyclic peptide, everybody is aware of the peptide bond. It is a covalent chemical bond between two molecules where carboxyl group and amino Similarly, peptide chain is formed when more than two amino acids are condensed together, and in real sense, when a tripeptide is formed, a dipeptide is coupled with the amino react together and with the release of water. Something regarding the history of peptide. Peptide is now a new word to us, as we are aware of essentially hormone is a peptide, and it was introduced in the clinic in 1920, followed by NTTS, calcitonin, oxytocin, the natural, up to calcitonin, the natural molecule, and the synthetic analogs were introduced in clinic from 1960 to 84, which are oxytocin, vasopressin, leuprolin, etc. there may be the heterocyclic amino acid like proline, histidine, tryptophan. Histidine is with the imidazole moiety, tryptophan is the indole moiety. And respective examples where these moieties can present are cited below, like in cyclopeptide, cyclopeptide B, sarcodactylamide by green arrows, proline moiety is indicated. among these cyclopeptide and sarcodactylamide are the Here is one slide showing the proliferating cyclic peptides, so which are biologically important. They may contain 1 to 3 amino acids avoiding, especially proline with trans and cis defect, as is indicated by different systems. There may be a single moiety with cis, single moiety with trans geometry, and there may be a single molecule cis and trans, and both trans moiety may be present. So these factors may affect the biological activity. There are some examples with cis and trans category of proline present in the cyclopeptide molecule. Both trans. Other moieties like halogenated bromo and hydroxy tryptophan isosirine dehydrohomoallanine may be present, like in cyclopeptide, gramamide, galactamide, as indicated by the arrow. Peptide structure. If you see the peptide structure broadly, they may be linear or cyclic. Here are some examples. Leopeptin, TRN, which is thyrotropin releasing hormone, and glutathione, all are linear peptide molecules. Here is a mixed molecule, antidiuretic hormone, vasopressin. It has a tail proline arginine glycine, and another part is hexapeptide molecule. Gramicidin S, which is a peptide antibiotic, is a pure cyclopeptide, and it has a 10 unit, so it is a decapeptide. Viroidin is an anti-tumor cyclopeptide. So related to sources, marine are abundant sources of cyclopeptides. Microscleroderma, Dyspodermia, Candela, Hymeniocidal species. The marine mollusks are another source of the cyclopeptide. This compound was isolated from Hymeniocidal species and synthesized in our organism. And it was hymenamide. Other sources are cyanobacteria, hypomycin, fungi, bacteria and plants. Here is the structure of the plant-derived cyclopeptide ICIC. It is isolated from diazoxide. It is the synthesized molecule in our life. Bioactivity of peptides mostly are associated with the cytotoxic activity. Although literature has not indicated that they are for the lung-specific cancer, but they are with other type of cancers they are associated. And the molecules which we research are tyrosin, samocylidine, sagatine. Here are the structures and their respective publications. Tyrosin 1 from the marine spore, samocylidine A from the roots of higher plant, and sagatine E from the seeds of higher plant, Basaria sagatine, and their respective publications. Anti-HIV activity is also there in the sertraline, indolecidine, calipaldin. Calipaldin and the indolecidine are the linear peptides, whereas cyclopeptide, which is associated with the immunosuppressive activity, is a cyclic peptide from flax seeds. It is cyclic disease. Antifungal activity, mostly they are active with the candida and the cryptococcus species. Halonitorolene, hazardamide are the molecules of interest as per our laboratory which is of marine origin, and hymenamide E, which is from marine spawns, hymen acidose species. Anti-dermatophyte activity against cryptophyton, metagrophytes and microspore molecules of interest. As per our laboratory point of view, sanitary C, styrene D, and 4D peptide is the sanitary C structure which is isolated from seeds of iron plants, as well as styrene G which is isolated from styrene comparison and their respective functions. Anti-malarial activity is type of structures are associated where alpha cyclopeptide A and B are cyclopeptides. Antibacterial activity, tyrosine 2 and cyclopeptide G. Tyrosine 2 is isolated from the microspores Indo-Caribean, whereas cyclopeptide G from seeds of inauguration. Anthelmintic activity, with anthelmintic activity, cyclic A, cyclic B, there are structures and respective application. Both compounds are cyclic in nature and isolated from latex of plants. These are some other type of activities associated with cyclopeptides. How they act? The literature review states that cyclopeptides act as cyclopsin by inducing apoptosis, which is programmed cell death. And here binding of IFG1 receptor is seen and antagonism of transport protein TGP and MRT1 are other vital mechanisms. Antifungal mechanism involves inhibition of cell wall biosynthesis via non-complicated inhibition of enzyme beta-1,3-D which forms the stabilizing glucan polymers from the cell wall. Antibacterial action, there is interference with the cytoplasmic membrane barrier as well as inhibition of transfer of peptidoglycan. Let's see the following diagram. So, cyclic peptide here binds with the LPS, lipopolysaccharide, and disrupts the outer membrane. So, overall, dearrangement of cell membrane takes place. There is increase in the permeability of cell envelope as well as leakage of the cell content and finally the cell. So, this is regarding the gram-negative bacteria. In case of gram-positive bacteria, cyclic peptide form complex with pectoprenol, which is a hydrophobic alcohol that plays key role in the growth of cell wall of gram-positive bacteria. And there is interference with dephosphorylation as well as shunt formation of bacterial cell wall. Peptides are good alternatives to small synthetic molecules. That's the question of interest. Why? As compared to small synthetic molecules, peptide possess less toxins and they do not accumulate in the water. Second fact is peptides are less harmful because they will disappear rapidly by proteolytic degradation and the degraded products are simply amino acids which have no toxins. So, as all of us know, peptide is body is a very weak bond and it will break. Peptides can work on their target very selectively, that is another advantage of the synthetic molecule. Comparison of linear and cyclic. If we compare both the molecules, cyclopeptides are always preferred over the linear peptide. Why? First reason is inherent flexibility of linear peptides because the chain is open. So it leads to different conformations which can bind to more than one receptor molecule and overall result will be undesirable adverse effects. That will not happen in the cyclic peptide molecule which have no freedom. Cyclization of peptides reduces degree of freedom to for each constituent within the ring, and thereby they will be reduced flexibility, increase potential selectivity of cyclopeptide molecules in comparison to linear. Another benefit is resistance to hydrolysis by exopeptidases due to lack of both amino and carboxyl as the compound is cyclic. So, cyclopeptide clinical trials, over 40 cyclic peptides are currently in clinical use, and it may be more than as the literature suggests, and around one new cyclic peptide drug enters the market every year on average. Approximately 140 peptide therapeutics are currently being evaluated in clinical trials, and mostly biotechnology industry they are working with. Here are some of the examples of cyclic peptide antibacterial drugs approved by FDA, like telemedicine, dalbavancin. These are for bacterial cell wall inhibitors and used for the skin infection and are administered as IV infusion. Here is the dosage form and the structure associated with these approved drugs. Another example of peptide-based drugs like exenatide, liraglutide, or the type 2 diabetes, econotide for the pain, and mifamotride for the bone cancer, dagarelix for the prostate cancer. All are peptide molecules if you will go through the structure. Cyclopeptide clinic or topical using felicitations, which is available as a topical cream which interferes with the cell wall synthesis, polymyxin B as cell membrane disruptor for the IBU, and like an immunosuppressive compound cyclosporic A, which is available as suspensible monoexoscylated capsule, which acts by lowering the activity of T cells and their immune response. Why peptide? Why not other molecules? Peptide market, if you will go to the peptide market, it is increasing day by day. You can see the example of Lupro from the Abbott laboratory in 2011. The sale went up to $2.3 billion. Lactose, another peptide molecule. It is actually a molecule at the border of peptide and a small bioparticle to the peptide bond. The sale reaches up to $7.9 billion. Lower graph clearly indicates US peptide therapeutic market size drug size from 2014 to 2025. It is increasing year by year. That indicates the need of peptides. How much you can suppose how much peptide drugs are being researched and are coming to the market. Global peptide therapeutic market and clinical pipeline inside 2026 report says global peptide therapeutic market opportunity for $65 billion. Among these, cancer peptide therapeutic market is worth $22 billion. Peptide drugs in clinical trials are 807. Clinical and patented site is 197 marketed peptides, and in pipeline there are 40. Just from the report to 46. Okay, this was the background where we studied what are peptides, what are their structures, from where they are isolated, and what is the market requirement, how we market the exposure is there towards the peptide market. So let's talk about how is this synthesized in laboratory. I am a peptide expert who deals with the synthesis peptide. So before going to the synthesis, we must consider the strength, weakness, opportunities, and strategies. So let's do what analysis. Like strengths are good efficacy, safety, shorter time to weaknesses are chemically and physically unstable, tendency for aggregation, opportunities are discovery of new peptides, alternative delivery routes, formulation development, etc. Price and reimbursement environment, increase safety and efficacy requirements for government, like that. These are the two basic reactions before going into the complex synthesis. When we start with the two amino acids, each amino acid has a 3 amino and a 3 carbon cell. And we need to protect one of the groups for a better company. So same thing, the first reaction is shown how a NH2 group can be protected. It can be protected by both. Both is the butyl oxy carbonyl. It is an anhydride. And these are the reaction conditions. And how to revert back the reaction by interaction with TFA, which is trifluoroacetic acid. So you will get the free amino group. You will react a both protected amino acid with trifluoroacetic acid. Then second reaction, how to protect the pre-carboxyl. It can be protected by esterification. So here you utilize the methanol and phenyl chloride, so it is methoxidase. Methyl group is introduced. And if you want to reverse the reaction, we have to do the alkaline hydrolysis with the least effect. So these two basic reactions are utilized in the whole peptide synthesis, and besides that, coupling is affected by a effective coupling agent. Here is the both protection mechanism. So, amide attacks the carbonyl side of the both, and tertiary carbonyl, butyl carbonate leaves as leaving, and finally the orbital, and then both protecting amino acid. In this, CO2 as well as the tertiary butanol is released as a byproduct. This is the deep protection mechanism where trifluoroacetic acid is used. If you go to the literature, you will see the other agents are also utilized for the protection. Like instead of both, you can use FMO, fluoride with carbonyl. CBZ is also used in the literature. So here you can see the difference between the two structures, which is boxglycine and F-boxglycine. F-boxglycine is associated with the aromatic, additional aromatic molecule. Coupling agent, and after proper protection, deprotection, coupling is required. So dicyclohexyl carbonamide is the most basic coupling agent used, but literature shows the use of PDC and PIC for very important. This is the coupling mechanism in these reactions that the DCU as a byproduct is formed. This is dicyclohexyl urea. So, okay. When we go for the routine coupling, sometimes problems persist. There may be formation of a oxazolone instead of a dipeptide bond, as is shown below by the middle structure. It is not our desired product, but during coupling, this product may form, and it is indicated when the final product IR spectrum will take as amide monoamide to form glutathione. Heterocyclic pads are used. So, to avoid this problem, racemization suppressing agents are included in the reaction. It is not necessary to add these agents, but sometimes if you better control the conditions, then we can avoid the use. So, what are the racemization suppressing agents effective in peptide synthesis? Of is the most commonly used, one-hydroxybenzodiazole, DMAP and anhydroxysulfide are the another substitute. formation of activated ester takes place. So here is the activated ester by interaction with the hydroxy benzyl. After reaction with the second amino acid, this group is released. So by this way, we can avoid the formation of zoonosis. Okay, let's consider a case of synthesized molecule in our laboratory, cysteine D, which is peptide. So, whenever to carry out such a compound synthesis, we have to go to the structure and we have to hypothetically break the structure on paper, not with a hammer. And then those units we have to synthesize in laboratory and we have to go for proper protection and reproduction and coupling and finally cyclization. So those are the simple steps. So here if you see compound number 1, it is protected at both ends, both and NS2 group is protected with both and carboxyl group is protected with the ester. And another is the amino acid molecule. So for coupling, because here we have the NS2 group. So we want COH group 3 here, so we have to do the de-esteric case using lithium hydroxide. So this OH group is free to couple with NH2, and when we will run the coupling reaction in the presence of these carbonamide, we will get a tripeptide molecule from a dipeptide and amino acid couple. So this compound is tryptophan, proline, glycine, methyl. Here, like different 3 and 4, both are dipeptides instead of amino acid. Again, one group, the COH group we need 3, one NS group we need 3. So both groups, here de-esterification is required using lithium hydroxide, and here deprotection of both is required using trichloroethylene. So when these both groups will be deprotected, it will be coupled to get a tetrapeptide group, which is both glycine, serine, leucine, methyl. Now we have two molecules, tripeptide and tetrapeptide, and we know we have to synthesize a heptapeptide. But before going for cycloheptapeptide molecule, we have to synthesize a linear heptapeptide. So these two compounds will be now coupled together, but before coupling, we have to deprotect at the proper end, like here. The tripeptide will be deprotected by de-esterification, and tetrapeptide will be deprotected at the pocket. And then it will be coupled together in linear heptapeptide molecule. But this is not our desired molecule, we need the cyclized molecule. So finally cyclization step takes place when it takes place in 4 steps. First, deprotection of the linear peptide molecule at carboxylate, so we will remove this methyl ester. It will be replaced with a para-nitrophilane or pentafluorophilane ester, which is a better leaving group. Then deprotection of the linear peptide at amino terminal here, and finally cyclization of linear peptide in the presence of space, where we keep the mixture for the 7 days at 0 degrees Celsius, and finally we check the band of free NH2 or COH group which disappears. Another cyclopeptide molecule, which is an N-methylated cyclopeptide synthesized, which can be synthesized for a cyclization step into. Here is the pharma spectrum of the molecule for alpha cyclic A. So we identified 4 types of fragmentation patterns which show the respective peaks in the pharma spectrum. So here, for example, A, if cleavage occurs at isomeristic theory and the component For the peak appears at 284, 355, 353. So here this is shown by the black arrow. These blue arrows indicate the ammonium ions which are due to the respective effluent in presence, like 120 is nitrogen, 86 is isolated reduces, 74 is purine, 72 is valine, and 30 is sucrose. These are specific for these elements. Another pattern you can see at cleavage at purine glycine end. And here we get the 357, 385, 456, 484 as indicated by the region. You can hypothetically break the molecule at different points and then can check for the particular peak present in the mass spectrum. Here is the summary of synthesized compounds from 2006 to up till now, which are antibacterial and antifungal in nature, like niacinamide E, anti-fungal actin, all compounds are synthesized in our laboratory. Peptides and peptides with antifungal activity and cyclopeptides, candidates and mechanisms, trichofibrone, mecanocyte, di-NCA, another cyclopeptide molecule, peptides, which is cyclotetrapeptide molecule, and cyclopeptide, which is antibacterial activity against the gram-negative. These are some of the enzymatic peptides synthesized in our lab: sagatinin B, long calicylin A, cerebellum, cyclopeptide C, gypsum B, cyclotetrapeptide. These all are published in literature. Some cytotoxic cyclic peptide molecules: cerebellum cyclopeptide E, sagatinin E, sagatinin C, cyclopeptide, cyclopeptide A, cyclopeptide. These are fluid cytotoxicity and pH. These are some of the newly synthesized cyclopeptides which we got between 2016 to 2023 when I left India and I came to Australia. This design of products as well as design of mono, di-halogenated and cyclopeptide. This is the scientific output in form of patent. We have a German patent, Australian patent. We have book chapters in collaboration with different authors. Article published in collaboration with different researchers. Professor Book Prize is one of them. The all are those papers which I have shown in the structure, cyclopeptide molecules to the being of. We have a collaboration with School of Chemistry, Mobile University, Institute of Pharmacy, Institute of Technology, Sydney, Institute of South Florida, Tampa, Siddhartha University, Fairfax, CIMS University, Malaysia, Institute of Sofia, Molega University, Jakarta, Sofia, University, Jaipur University, and University. We decided doing research here, we are well involved in the community outreach because we run a PhD pharmacy 4-year program and which is like a pharmacy. It is not a pharmacy, but it is like a pharmacy. They go in hospitalized community pharmacies and open open retail pharmacies. They don't have much he usually goes on the World Health Day or World Health Organization approved day so that we can educate the public. Like today we are doing our World Lung Cancer Day, through this conference we are educating our pharmacy students around the globe. So we used to visit in different schools and give them basic and some of the things to avoid at the age, at the teenage so that they can be habitual of those studies. My lab visitors, our research group, some of our colleagues who went to our field. Here in Trinidad we are almost 50% black population and 50% of the Trini Indians. Forefathers came here and settled. They do not speak Hindi. Their forefathers spoke Hindi and now they forgot it. English is the national language. Here are some of the things. My name came under the Stanford University 2% scientist. You know, one another fellow, Professor Gupta, who is also studies. So this is Caribbean Association of Pharmacists. I was selected as a distinguished pharmacy researcher. I got the Productive Researcher Award for Scientific from Faculty of Medical Science. And this is our cancer principal who celebrated the day when we were selected for the among the two pharmaceutical science. Here, for a comparison, India and Trinidad. Trinidad is a small country, one island, twin island you can say, with a population of 1.4 million. There it is double with a capacity to like our Chole Bhature. Then we have the dance competition, carnival, we have beautiful beaches, and we have our university on the island. There is only one school of pharmacy with the 60 capacity in the country, where I remained as director for 6 years. Now I am serving as professor, only professor of the island. So there is no other pharmacy institute. Only we educate at every pharmacist or we can give us. So that was about some research, basics of peptides as well as the opportunity for collaboration. And I have explained about Trinidad who want to visit, whether it's purpose is research or research. Thank you very much for your time. Email on my email. I will be happy to hear all the queries.
Thank you so much, sir. For the oral presentation, Sakshi Gupta, are you there? After Sakshi Gupta, Priya Bande, please be ready. Sakshi Gupta is from ICIC University.
हेलो, यहाँ पर सभी जो फार्मेसी की फैकल्टीज़ हैं उनसे रिक्वेस्ट है नीचे एडिशन सेल में सब लोग वहाँ पर गैदर हो जाए। प्रोफेसर दीपक कुमार सर आए हैं जो यहाँ पे की स्पीकर, वो थोड़ा फैकल्टी से इंटरेक्ट करेंगे, कुछ बताना चाहेंगे। थैंक यू वेरी मच।
showing the acridine compound. So, if we talk about acridine, then it is a very versatile molecule as well as like it is showing various activities like it is showing the multiple activities that is antimicrobial, anti-cyclopeptides. Drugs are there for the malaria and for the cancer, which is based on the acridine pharmacophore. So these are some examples which are even marketed. That is amsroquine. It is an example for the anti-malarial drug, and as well as it is using for the anti-cancer. Nitrocrete is also used as an anti-cancer drug. If we talk about naproxen, it is used in the various inflammatory diseases. And if we talk about pyronoacridine, it is used for the malaria. So, with the help of this, I just reviewed the various articles, due to which I came to know that acridine is very versatile molecule and it will be used for the further derivatization, through which we can design various new or novel analogs. With the help of, like, these are the three basic reactions, that is Ullmann reaction, Van-Ryden reaction and Friedlander reaction. These are the basic reactions which is used to design the acridine containing moieties. So basically this is the example for the Ullmann synthesis reaction. In this, the starting material is anthralinic acid. And one more thing is here that anthralinic acid is nowadays banned in India. So we can just create that particular moiety, anthralinic acid, in the labs as well. And with the help of this anthralinic acid and the aryl halides, and with the help of aniline, we can create the acridine moiety in the labs, for which the different analogs is designed nowadays in the lab. Next reaction is the Bunsen reaction. In this, first the starting material is diphenylamine. With the help of reacting with the carboxylic acid, various carboxylic acid, we can design the 9 substituted acridine. Next reaction is Friedlander reaction. In this, we are using the acrylic acid, which will be treated with the cyclopeptides to give the acridine compounds. And the next reaction is from the acylated diphenylamines. When we react the diphenylamines with the iodine, then we can also meet the acridine moieties in the lab. These are some pharmacological activities. As I have reviewed many articles, so these are few of them. Like I have
Hello. Priya, very nice presentation. Sakshi, very nice presentation. Activities. So, what do you mean by the term pharmacophore? You have written. What is the
Pharmacophore is a basic moiety on which we can attach the different substituents to design the various derivatives. Like a basic moiety, if we talk about the anthracene, so in that particular anthracene, at a position we can replace the carbon with the help of nitrogen, a heterocyclic compound, heterocyclic moiety. So if we talk about the acridine, that is a pharmacophore on which various attachments can be done to design the various analogs.
What is the meaning of pharmacophore?
So pharmacophore is a basic moiety, like a base
Basic
moiety.
moiety means what? What do you mean by the term moiety?
So moiety, like the structure.
Yes.
Yes.
So pharmacophore is a structure.
Yes.
Basic moiety or structure. Very good. Can I see the acridine moiety? Acridine structure.
Yes.
So what is acridine moiety? This is a tricyclic structure.
Yes.
So how do you predict the features of pharmacophore? How do you predict the essential features? You have told that it has so many activities like antimicrobial.
Yes, so nitrogen is basically a molecule which can depart space. So if we do work on any heterocyclic compounds, then it will be helpful for the various activities.
No, you see the structure.
Yes, sir.
You look the structure.
Yes, sir.
You look the structure, and you tell me what features it has. That is the pharmacophoric feature. It is aromatic.
Yes, sir.
So it produces aromaticity. Aromaticity, one of the feature of pharmacophore.
Yes.
Nitrogen is there.
Nitrogen is there.
It can contribute for
So various pharmacological activities.
means in terms of chemical properties.
It can depart necessity to the compound.
Hydrogen bonding interaction.
Yes, sir.
Yeah. These are the two important. Very nice. God bless you.
Thank you, sir. So much. Next participant is Priya Pandey from Devasthi Vidyapith College of Pharmacy. Firstly, I am thankful to Devasthi Vidyapith College of Pharmacy that they have given me that they had organized such a
nice presentation, Priya. This is your basic concept you have got, or what original research?
Sir, actually this is a,
What is the drug name you don't want to disclose?
sir, this is topical.
Means drug you don't want to disclose. Oh, very nice. ये nano formulation scientists, they are trying, they are telling, they are shouting that we are the best, tablet capsule emulsion, suspension fail, nano formulation is the best one.
Yes, sir.
So you also agree with that, right?
Yes, sir.
Very good, very good. ये cubosome, what is the normal particle size as per X-ray diffraction?
Sir, for penetration at the skin surface, the normal size will be the less than the 500 Dalton. Sir, but the cubosome size is 10 to 500.
Dalton? Nanometer. 10 to 500 nanometer. Are you sure?
The size range of cubosome is 10 to.
Okay, you check. I think I am down. You check once. Okay. And then you have written amphiphilic, hydrophilic, hydrophobic. So these are the drugs nature or what? Means overall nano formulation nature, cubosome nature, or particular API nature. The drug which you try to produce the nano cubosome.
Sir,
Drug that you want to formulate.
sir, the nature of the cubosome that it can encapsulate hydrophobic, amphiphilic, and hydrophilic, all types of the drugs. It can encapsulate and deliver to the target site.
Okay. Very good. Any other question?
Very good presentation.
Thank you.
What is the zeta potential of cubosome?
Sir, approximately, sir, plus minus 20 megavolt.
In between
20 to 25 megavolt.
20 to 25, are you sure? What is the unit?
Sir, megavolt.
Sorry? Unit. Zeta potential.
Sir, megavolt.
Okay, you can check. Thank you.
Thank you, sir. Thank you so much. Now it is time to honor some esteemed guests for today. I would like to invite Dr. Cecil Nandi, sir, Professor at Global Institute of Pharmaceutical Education and Research, Kashmir, and I request our convener, Professor Dr. Manoj Bisser, to please hand over the token of gratitude to our guest. Come to the stage. Thank you so much, sir. Please, I request Mr. Amit Sen, sir, to come to the stage. Amit, sir, is from Samrat Prithviraj Chauhan College of Pharmacy, Kashmir. Thank you so much, sir. I request our organizing secretary, Mr. Manoj Bhadwa, sir, to please present token of gratitude.
Mr. Himanshu Kohli, Samrat Prithviraj Chauhan, College of Pharmacy, Kashmir. So here we come to the most awaited part of this conference, the prize distribution ceremony. I'm going to announce the name of the winners of poster competition, and the first position is secured by Nirmal Joshi, Faculty of Pharmaceutical Sciences, Amrapali, Khalwani. Now I would like to request our convener sir to kindly give the award to Mr. Nirmal Joshi.
सेंटर
The second position is backed by Sandhya Datia, M.Pharm student from Global Institute of Pharmaceutical Education and Research. I would like to call upon Mr. Bhardwaj sir to give the certificate and memento. Many congratulations. Third position is backed by Nikita Balkoti, M.Pharma student from Department of Pharmaceutical Sciences, Bhimtal Campus, Kumaon University. May I call upon on stage Dheeraj Nautiyal sir to offer the memento and certificate. Now we move on to the winners of oral presentation. The first position is backed by Sakshi Gupta, IFTM, Moradabad. I would like to call upon Dheeraj Bish sir to do the honors.
आज के इस इंटरनेशनल कॉन्फ्रेंस में पुनः के लिए आपका स्वागत है। सबसे पहले मैं स्पेशल थैंक्स बोलूँगा जो हमारे की स्पीकर्स हैं, डॉक्टर गौरव गुप्ता सर, डॉक्टर दीपक कुमार सर और प्रोफेसर डॉक्टर राजीव अय्यर सर। इन्होंने अपना वैल्यूएबल टाइम हमको दिया, जबकि ये बहुत बड़े इंडियन स्पीकर्स हैं, उसके बावजूद भी ये हमारे इवेंट का पार्ट बने। और इसके अलावा मैं देवस्थी परिवार की ओर से भी सभी को धन्यवाद दूंगा कि सभी लोगों ने अपनी सहभागिता इसमें निभाई और सभी ने इसको अपने बिल्कुल जैसे एक काम होता है, घर का काम होता है, उसी ढंग से इस पर लगे और कहीं पे कोई कमी नहीं छोड़ी। कहीं पे किसी ने कोई भी अपनी ओर से ऐसा लगा कि हमें कोई चीज़ लैग कर रहे हैं या कुछ छोड़ रहा है, तो मैं स्पेशली देवस्थी परिवार की, देवस्थी DVIP की और DPCP की फैकल्टी को भी स्पेशली थैंक्स देता हूँ कि उन्होंने बहुत ही कॉर्डियल और बहुत ही कोऑपरेटिव तरीके से इस इंटरनेशनल सेमिनार को संपन्न किया, इसको सफल किया। और मैं इसके अलावा हमारे मैनेजिंग डायरेक्टर सर श्री वनिंद कोश्यारी सर, एग्जीक्यूटिव डायरेक्टर सर श्री रमेश चौधरी सर, कैंपस डायरेक्टर सर और उसके अलावा हमारे प्रिंसिपल सर डॉक्टर मनोज बिष्ट जी, इनको भी स्पेशली थैंक्स देता हूँ कि ये भी दिन रात एक करते रहे। स्पेशली मनोज बिष्ट सर को भी थैंक्स देता हूँ कि वो कल रात के 11 बजे यहाँ से गए हैं और जब तक ये काम उन्होंने पूरा नहीं किया उन्होंने अपना रोल पूरा इसमें प्ले किया। और इसके अलावा जो हमारे स्टूडेंट्स हैं, जो हमारे स्टूडेंट वॉलंटियर्स हैं, उन्होंने भी अपना इसमें 100% योगदान दिया। मैं अपने स्टूडेंट्स को भी स्पेशली इसके लिए थैंक यू बोलता हूँ क्योंकि बिना स्टूडेंट्स के लिए चीज़ें संभव नहीं थी और स्टूडेंट्स को भी स्पेशली मैं थैंक्स देता हूँ। और इसके अलावा मैं प्रोफेसर दीपक कुमार, इनको तो मैं स्पेशली थैंक्स कहता हूँ क्योंकि ये नेशनल साइंस कांग्रेस जो कि दिल्ली में कल थी, उनकी एक मीटिंग थी, क्योंकि सर एक अपने आप में मैंने अभी जैसे बताया था नीचे डिस्कशन में कि बहुत बड़ी इमिनेंट पर्सनैलिटी हैं, बहुत बड़ी नॉन पर्सनैलिटी हैं, उनके करीब 55 पेटेंट्स हैं, उनके 25% जो पेटेंट्स हैं वो तो ग्रांट्स हुए हैं और उनका नेचर में भी उनका पेपर पब्लिश है। नेचर में कोई आज ये बात बताओ एक चीज़, आप इस पर चाहे यकीन करें ना करें, ये एक ऐसे स्पीकर हैं जो इस हमारे कुमाऊं की बेल्ट में, इस पूरे उत्तराखंड की बेल्ट में पहली बार एक ऐसे साइंटिस्ट यहाँ पे आए हैं जिनका पेपर नेचर में है और जिसका इम्पैक्ट फैक्टर 44.5 है। इसके लिए एक विशेष क्लाउड और विशेष तालियां होनी चाहिए। तालियों में थोड़ा सा कमी है, एक जोरदार तालियां दीपक सर के लिए बहुत, और ये हमारे बहुत अच्छी बात है। और अभी दीपक सर ने नीचे फैकल्टी से भी इंटरेक्ट किया, थोड़ा समय का अभाव था, थोड़ा समय कम था, लेकिन हमारे डायरेक्टर सर ने कहा है कि अक्टूबर के मंथ में जब थोड़ा सा हमारा कोर्स पूरा हो जाएगा, सर यहाँ पे आएंगे और सारी फैकल्टी से इंडिविजुअली इंटरेक्ट करेंगे। और हम इस बार नेक की ओर जा रहे हैं, तो हमारा एक टारगेट है कि हम 25 से 30 पब्लिकेशंस, ऐसा नहीं कि अब तक तो क्या था कि कोई भी दे दिया, अच्छे पब्लिकेशंस में, अच्छे ब्रांड में, जैसे कि एल्सेवियर है, विली ऑनलाइन लाइब्रेरी है, बेंथम साइंस है, टेलर फ्रांसिस है, अमेरिकन कोरियन सोसाइटी है, रॉयल कैपिटल सोसाइटी है। क्योंकि सर कहते हैं कि ब्रांडेड में ही काम करो, कि पेपर आप साल में एक लाओ, लेकिन वो पेपर ऐसा हो जो फार्मेसी में तहलका मचा दे, जिसको साइट करे। और सर का तो H इंडेक्स और I इंडेक्स भी कम से कम 40-50 के पार है, जबकि एक और सर का भी टारगेट है कि यहाँ पे जो फैकल्टी है DPCP की, हमारा अभी H इंडेक्स I इंडेक्स आने वाले 2-3 साल में 10 पार तो हो जाए। अभी तो सोचता हूँ कि शायद हम 5 तक भी पार नहीं हुए होंगे, लेकिन अगर सर का हमें पूरा कॉपरेशन रहेगा, सर की हमें गाइडेंस रहेगी, तो हम वहाँ पर भी जरूर उस ऊंचाई को भी छुएंगे जब वहाँ तक पहुँचेंगे। तो मैं स्पेशली प्रोफेसर दीपक कुमार सर को थैंक कहूँगा कि उन्होंने अपनी वैल्यूबल मीटिंग को भी छोड़ते हुए मेरे आग्रह पे, हमारे DPCP परिवार के आग्रह पे वो यहाँ पे आए, अपना समय दिया। और पुनः मैं ज़्यादा बात न करते हुए सभी हमारे जो यहाँ पर स्टूडेंट वॉलंटियर्स हैं, सभी फैकल्टी मेंबर्स हैं, प्रिंसिपल सर हैं, मैनेजिंग डायरेक्टर सर हैं, एग्जीक्यूटिव डायरेक्टर सर हैं, हमारे चीफ ग्रुप एडवाइजर भंडारी सर हैं और सभी फैकल्टीज का मैं स्पेशली धन्यवाद देता हूँ। और हम आगे भी ऐसे आयोजन टाइम पे करते रहेंगे और इससे भी हम बहुत अच्छा आयोजन करेंगे। और प्रोफेसर दीपक कुमार सर से रिक्वेस्ट करता हूँ कि हमारी संस्था से वो हमेशा जुड़ें और हमें अपनी गाइडेंस हमेशा प्रॉपर्ली एक बड़े लेवल पर देते रहें। बहुत बहुत धन्यवाद। थैंक यू वेरी मच।
सारे आ रहे हैं, सारे आ रहे हैं।
सर आप लोग फिर थोड़ा सा आगे आ जाइए। गुलदस्ते तो नहीं आ रहे?
रहने दो, नहीं आ रहा।
इधर देखिएगा।